Okay, we're going to pick up where we left off and add a little bit of confusion. Respiration. In biology, it has a particular meaning that's different than breathing. Respiration is a process where you take big molecules, you pull them apart, you take the energy that was holding them together, and you use that to build more usable molecules. Usually a molecule called ATP, which is unstable enough to be really useful for making stuff run, and stable enough that you can move it around in a cell. You can't circulate in the body, it's not that stable, but you move it around in a cell and run things off of it. Now, prior to oxygen being available, there already would have been many, many, many different forms of respiration. We still have many, many forms of what's called anaerobic respiration, respiration without oxygen. But it turns out that oxygen is a much more efficient end grabber at pulling more and more energy out of the molecules you're pulling apart. You get way more ATP molecules using oxygen than doing the other types of respiration. So some organisms evolved systems to do that, they already had been using things like sulfur and iron, other sorts of things as a kind of an end grabber. And so moving oxygen in there was a bit of a leap, but not a huge leap. And now you have aerobic respiration, much more efficient. Now we're still in the prokaryotes though. We have prokaryotic photosynthetic organisms and prokaryotic aerobic organisms. Eukaryotes, bigger cells, separate internal chambers for doing different sorts of jobs. That's very efficient. A chamber just for processing your DNA. But it turns out some of the chambers that you find in eukaryotes didn't actually evolve as chambers in eukaryotes. They were kidnap victims. A researcher back in the 1960s who was interested in how aerobic respiration had evolved started to look at bacteria that could do it, looking to break down the chemistry and compare it to the chemistry in eukaryotes. Well, first off, she found the chemistry was close to identical. And then she started to notice that these prokaryotes that do aerobic synthesis, respiration, are almost identical physically to the chambers in eukaryotes called mitochondria that do aerobic respiration. And she published papers on it, and people went, "Oh, Lynn, that's kind of crazy that, cells took in little cells, and instead of digesting them for food, they actually held onto them because they were useful. And then they would pass them from cell to cell when the cells reproduced, and the little things would reproduce inside the cells. That's a crazy idea." And it was kind of a brand new kind of, crazy idea, but it probably didn't help that it was the 1960s and she was a woman, and women really hadn't really gotten to a point where they were as accepted as the guys. In biology, there's actually more women in biology now than men. Some of the other sciences like physics and to some extent chemistry, that's still an uphill climb if you're a woman, you're trying to get people to listen to you. So she published other papers going, look at this structure that's the same, look at this structure that's the same. And they went, the way science is supposed to change, but sometimes you got to really give somebody a kick in the pants to make them change their opinion. Here's what she found. Prokaryotes have different types of chromosomes than eukaryotes. Prokaryotes have little loops, just a single little loop in a bacterium is their DNA. Eukaryotes have matched pairs, right? We did this in genetics. They have matched pairs, which allows for sexual reproduction. That's a great thing. But the chromosomes have two ends to them. They're not loops. Guess what you find in mitochondria if you look close enough? Little loop prokaryote chromosomes. Not enough to build a whole mitochondrion, but genes that are only used in the mitochondrion are in there on the loop. Over the course of billions of years of being residents in the bigger cells, a lot of the genes that could do other stuff in the cell had been kind of ferried out to the nucleus, because the nucleus processes DNA better. But the mitochondria had held on to a few key genes, and it was a bacterial, a prokaryote chromosome. That was kind of the slam dunk, as people went, okay, this happens. An idea is really iffy. People push against it, push against it, push against it. Eventually you find just the one little detail that gets them to go, oh, okay. Yeah, we knew it all along. After that, she looked in plants at the organelles, the little chambers that do photosynthesis. And she found that they exactly matched a type of single-cell prokaryotic algae. And when she looked closely enough, she found little prokaryote chromosomes in there too. So now the accepted idea is that these two chambers in our cells started as independent bacteria that were taken in for food, but were actually more useful to keep alive because they did this great thing. They used oxygen in an efficient way. They made food with using sunlight as an energy source. And they were so useful to keep around that they eventually just became permanent residents, and it wasn't even that noticeable, that recognizable that they ever were separate. Now, since then, we have found this happens today. Even around here in the ponds, there are single-celled eukaryotes that gobble up algae and just hang on to them, use them for photosynthesis. And this time of year, it's wintertime. If we get enough of a snow cover that the light doesn't make it down to where these guys live, then there's no light, there's no photosynthesis. They go, well, heck with this, and they just gobble up the little kidnap victims that they have, and they'll pull in more when the spring comes. So it just shows as a proof of concept that this can happen, and they found it a bunch of other ways where bigger cells will take in little cells and use them rather than just make food out of them. Now we've got eukaryotes. Another thing that's a great thing that eukaryotes can do, that prokaryotes can't do, is that they can work together as systems. The only multi-celled systems are eukaryotic systems. You can find a little bit of colonial activity in some bacteria where they get together and they're not all doing exactly the same thing. Some of them are kind of cooperating to keep the whole colony going, but it tends to be very simple and it's kind of rare. Where in single-celled eukaryotes, there's a whole bunch of different colonial single-celled eukaryotes. And that's just a step away from being multi-celled. In fact, there's another step in between. There's some types of sponges, which are multi-celled animals, where if you take the sponge and you put it in a blender and you hit it at the right setting, it'll pull the sponge completely apart, pull all the cells apart without killing them. And if you take that soup that you get and pour it into a tank, they cannot live independently for very long, but they will find each other and reform the sponge that you pulled apart. If we put you in a blender, you're not going to do that. And very few multi-celled, legitimately multi-celled systems can do that. But colonial systems can. And in fact, they can happily, not so happily, but they can live on their own as separate cells if they absolutely need to. Just normally they don't do that. Another big deal with eukaryotes, as I mentioned before, sexual reproduction, a new way of combining features, successful features, as evolution goes along. I'm going to mention in passing something that they call the Gaia hypothesis. This is the idea that the Earth itself, once you've got an oxygen atmosphere and water cycles and other kinds of things, that the system itself becomes hard to destabilize. That as potential climate changing systems, conditions move, is that it's hard to shift the climate too far in a particular direction. You can shift it far enough that you kill off a lot of things, but not enough that you kill off everything. The Earth is like a self-regulating system. Gaia was a Greek goddess of the Earth, which is where the name comes from. Not an easy thing to test. People think they see it in the history. Okay, I'm going to skip ahead, but I'm not going to write anything down. You'll see why in a second. There are a few fossil beds from a long time ago. There's no real animal fossils, or very little in the way of signs of animals. There's nothing, there's nothing, there's nothing. And then suddenly there's not just animal fossils, but you can find almost every major phylum that we're going to talk about in the second half of the semester represented in these fossils. You can find members of our phylum, you can find members of the arthropods, you can find annelids, you can find mollusks, all the big guys. You can find things that you can recognizably put into that phylum. This little guy here would have been related to us. So, nothing looks like it, a modern fish, but something with the right kind of features to be in the chordates, which is our phylum. And it appears for fossil purposes fairly suddenly over the course of just a few tens of millions of years. And they call that the Cambrian explosion. Cambrian is the name of this period in history, and kind of an explosion of semi-modern forms kind of all at once. And the question always was, why? Things had been happily scooting along for 3 billion years without a heck of a lot happening, and suddenly in a very short period of time, we get all of this. And nobody really knew. And we have another story like the endosymbiont story. A geologist is looking at sedimentary rock from a period before this happened. Now, sedimentary rock is the holdover of ocean sediments. At least what he was looking at was ocean sediments. And he's looking at these layers, and he would find these big rocks just embedded. And he's, uniformitarianism. If this was modern times, what would that rock be? Well, he knew what the rock would be. It would be what's called a drop. You see these around here, right? You get up into the woods and you see these big rocks just kind of sitting on the surface that don't really match the rocks around there, and they're separate. They're sitting on the surface. Glaciers. This used to be covered with ice. Right where I'm standing used to be under like a half a mile of ice. And the glaciers move. They move downhill. And like big snowplows, they push stuff in front of them and they pick stuff up from underneath. Is that they integrate big old rocks into the ice as the ice moves along. When glaciers reach the ocean, they keep getting pushed from behind, big chunks drop off and become icebergs. The icebergs have these rocks embedded in them. Around here, when the glaciers retreated, when things warmed up, they would drop the rocks as they melted. Same thing happens in icebergs. As they drift, the water isn't freezing, it's a little warmer than they are. As they go, if they drift further south, it gets even warmer, and they melt and they drop the rocks. And a guy goes, this is an iceberg drop. Okay, interesting. Here was the kicker. The sediments he was looking at were formed on the equator, right in the hottest part of the tropics. Well, icebergs don't get anywhere near the tropics. They melt way before they get anywhere near the tropics, and this would have been true unless it would have been a heck of a lot colder during this ancient period of time than it is today. And so he published a paper on it going, wow, it looks like there was a huge ice age we didn't even know about. And people went, no. The continents move around. Is it this layer you're looking at? Yeah, it was pretty close to the equator when you looked at it, but it wasn't close to the equator when it formed. And he went, no, I think it was. And he did some follow-ups. And you can actually, by looking at the magnetism in some of the little bits and pieces of the sediments, you can tell where it was on the planet when the sediments settled. And so you can get a location. north-south location. And from that, he was able to publish another paper going, nope, look, this was on the equator. Is that the way that the magnetic lines work, this was formed on the equator. There were icebergs still floating in the waters in the equator. People, oh, wow. And you know what also often happens? People went, oh, you know, now that you mention it, there's some other stuff we were having trouble explaining, and this makes a lot of sense now. They started to look into it. Now, from other evidence accumulated, it was not just an ice age, it was a major, major ice age. It had lasted for millions of years. At one point, the entire Earth was covered in ice except for some of the places where there was volcanic activity, some of the surf zones on the tropics. But in the middle of the oceans in the tropics, there may have been as much as a half a mile of ice on the surface of the ocean. Bottom of the ocean was still liquid. And some other areas where there was volcanic activity was still liquid. But the number of places where now life existed, but the available real estate really shrank and the conditions changed in a major league way. Competition and changes of conditions is what pushes evolution, right? Where did the Cambrian explosion come from? From this period of intense, limited real estate competition. Is that the Earth eventually, because of volcanic gases, it warmed back up, the ice melted back. Is that from these little places around the planet, things that had evolved there, fighting for life during the snowball period, spread out through the warmer Earth, and eventually wound up in that layer that I showed you of all those different animals with all sorts of new hard parts and teeth and shells and all sorts of protection from, things trying to chow down on them because there wouldn't have been a heck of a lot of plants. So that's the Cambrian explosion, and that explains where it came from. Another thing to keep in mind, these are great moments in evolution. We still have like 250 million years to go before we find much in the way of evidence that the continents had anything alive on them. There were still continents out of the water, but life was in the water. And then eventually we start to see evidence of what are recognizable land animals and remnants of land plants. So that's the next big stage in the history of life. Now, there would have been continents, and there would have been weather, and there would have been rivers and outflow from the continents, and the rivers would have picked up eroded minerals and other kinds of things that are useful for living things to have. So life would have existed on the fringes of this. And in order to live in tidal pools, in order to move up the rivers and get into smaller and smaller moving streams, there are some adjustments you need to make from living in the oceans that kind of set you up to be able to live in the air on land. And let me go down the challenges. Okay, you can write these down. When you're surrounded by water, you don't need to worry about drying out. When you're out in the air, you're going to lose water to the circumstances. Now, a lot of animals in tidal pools have shells or exoskeletons. That's where those land animals probably started. You got to scurry from tidal pool to tidal pool. Also, it rains into the pools. the pools dry up is that when the surroundings, when you're in the water and the surroundings get more dilute, that's a problem because water will come into your cells and swallow them up and pop them. When the water gets less dilute, you're going to lose water to the surroundings and your chemistry is going to get messed up. Those are both issues in tidal pools and in fresh water. Fresh water is like 100% water. If you live in 100% water, it's going to flood into your cells and swallow them up and pop them. You can actually kill yourself by drinking too much water. Your kidneys are pretty good at keeping the water levels in your blood balanced with your cells, but if you throw too much water into your system, water floods into your cells, it swells up your brain cells. People have died from drinking too much water. But if you live in fresh water, you better be able to deal with it. And there's a couple of different ways to do that. The plants have a casing around their cells that keeps the cells from swelling up too much with extra water so they can't swell up and have problems. Animals typically either pump water out really fast, which is part of what our kidneys do, or the bigger animals moving in tidal pools and in fresh water waterproofed their outer surfaces. I know it's weird to think that fish are waterproof, but bony fish with bony skeletons, are largely waterproof, even the ones that went back into the oceans. When you're waterproof, water's not coming in through your skin in order to swell your cells up. You got to deal with it coming in through your gills because you got to let water in, because you got to let oxygen in. You got to come in through your digestive system, and your kidneys deal with that, pumping it out. But just dealing with those situations, if you're waterproof to keep water out, you're also waterproof to keep water in if you're out in the air. So drying is already kind of pre-adapted to. Support, right? Water helps hold you up. Is that getting out onto the land, you need to be able to hold yourself up. Well, scurrying around in fresh water, shallow, fresh moving water, or scurrying from tidal pool to tidal pool, or scurrying back to the ocean when your tidal pool dries up, those all require support in the air. So again, starting it out. Some stuff that you don't necessarily think about. Water doesn't change temperatures real fast unless it's fairly limited, or it's filling with rain out of the atmosphere. And so things that live in fairly deep water don't deal with a lot of temperature fluctuations, nothing fast. But in the air, you know from living around here, living almost anywhere, the air can change temperatures pretty doggone fast. But it can change pretty fast in a tidal pool or in outflow from rain as well. So again, those are good staging areas for that. Another thing you don't necessarily think of, sunlight can mess with chemistry. There's a lot of frequencies of light that can mess with chemistry. Water absorbs and reflects a lot of that. So unless you're right up at the surface all the time, you don't really need to deal with sunlight issues. But if you live in a tidal pool or you live in shallow fresh water, you're going to get exposed to a lot of direct sunlight effects. So that kind of predisposes you to get out onto the land. Another thing, we've been talking about oxygen building up. Water doesn't dissolve that much oxygen. A lot of the oxygen would have percolated out into the atmosphere and built up to much higher levels. Remember, oxygen's reactive, it can actually be poisonous. In order to be out in the air, these water organisms had to be able to deal with higher levels of oxygen. And in fact, they eventually made it work for them. More oxygen, more respiration, higher metabolism. There's a reason why land groups that have members that have gone back into the water, like the crocodiles, or the dolphins, or the penguins, those guys never evolved to breathe water because they're taking this high land metabolism into the water, and there's just not enough oxygen for them in the water. They got to come to the surface and get the oxygen from the air. Difference between about 6% and about 20%. That's a big difference in available oxygen. Things that have lived in the water permanently, some of them have some pretty high metabolisms. They get pretty efficient at using the oxygen available to them. But again, shallow fresh water, you're exposed to the air a lot. Tidal pools, you're exposed to the air a lot. Now, the last thing that applies to everything, there's another thing on the list, but it's the last thing that applies to everything, reproduction. Swimming sperm was the going thing in sexual reproduction for an awful lot of these organisms. They don't swim in the air very well. And what we'll find when we look at these groups is that reproduction was the very last problem to get solved by plants when they got up on land and by animals. Well, by our ancestor animals when they got up on land. Is that they came up with some, basically using water that was available for a long time before they came up with ways to get the sperm from where it's made to where it needs to be working through the air or injecting it directly into a female's body. One other thing on the list. Plants need nitrates to make amino acids, to make proteins, because proteins are the chemistry thing. There's nitrogen in the atmosphere, lots of it, but not a lot of available nitrates up on land. But in the surf zones and in the tidal pools, they'd already been partnering up with symbionts, again, bacteria and funguses that could pull nitrogen from the air and combine it and make nitrates for the plants. And that's what they needed to become land plants. And they still are. If you look out 90-some percent, probably 99% of the plants you look at and see have buddy organisms, bacteria and or funguses, that convert the nitrogen in the air to nitrates for them so they can build proteins and survive. They absolutely positively need these guys, often embedded in their roots, but sometimes in other parts of the plant. Where the plant gives them a great place to live and it gives them fuel, it does all the nice things that it does for the food chain, but it does it for these buddies, and it gets nitrates back from them. Okay, there's a very, very short ending to this chapter, but we're running pretty long, so I'm going to put a really short video on it.